Okay. So it's always difficult to predict the future in this session, but I'll talk about a sort of technological platform that I think is familiar to most people that's useful for analysis of coronaviruses, both current and future. So I've been working myself on coronaviruses for over 20 years, initially on some human coronaviruses. Then when I moved to Bristol, SARS-CoV, and then subsequently murine hepatitis virus, feline coronavirus, MERS coronavirus, and then of course SARS-CoV-2. At Bristol, I also work with David Matthews, who's started on MERS and SARS more recently. And a large focus of the work I've done for SARS-CoV, feline coronavirus, and SARS-CoV-2 has been focusing on reverse genetic systems for these viruses. So the question might be, these are quite obvious really, but why would you want to produce a reverse genetic system anyway? Well, the most obvious one is probably to investigate the effects of genome substitutions or mutations on virus phenotype using suitable models. Another one that maybe is less well understood by some people is production of, well, certainly reporter viruses, but what are known as replicon systems, where we disable the virus but allow it to replicate components of it in cells. So it's very useful for instance, antiviral research. And of course, production of live attenuated vaccines, which we haven't really seen so much with SARS-CoV-2, but has been certainly a very well-studied theme with animal and avian coronaviruses over the years. So compared to other RNA viruses, coronaviruses have been the most difficult to develop reverse genetic systems for. And that's for a number of reasons. First of all, regions of most coronaviruses or their genome, cloned as cDNA, are unstable in bacterial plasmids. So this has really hindered efforts. And just because of the large genome size, it's really necessitated sort of much greater sequencing efforts. And if you're dealing with a protocol that requires production of in vitro RNA transcripts of 30 kilobases, this is also really quite difficult. Over the years, there's been a number of different approaches. A number of these I've used. So the first of these was the use of a vaccinia virus as actually a cloning vector to help stabilize the cDNA clones. And I was fortunate to be sort of in the laboratory of Stuart Siddell working with Volker Thiel when they established this method. And subsequently, I used it with Volker and Stuart to produce a reverse genetic system for SARS-CoV. There's the system produced by Ralph Barrick in his laboratory, which was just ligation of cDNA clones independently spanning whole coronavirus genomes to produce full-length cDNAs. And this is one that we've used for feline coronavirus, which still is certainly not trivial by any means. And a method pioneered by Louis Njoanis, which was cloning the full-length genome in a BACMD vector, and sort of manipulating in bacteria, but still this has its problems with sort of instability as well. So at the beginning of the pandemic, I was in contact with Volker, and he told me about his new approach where he'd rapidly assembled overlapping cDNA clones in a YAC vector, and that he could, when this first system was established, it took over 3 years to develop reverse genetic system for coronaviruses, and now he could do this in less than 2 weeks. So we tried this for SARS-CoV-2, and one important point is even before the virus was in the UK, we'd ordered overlapping cDNA or overlapping gene fragments that were synthesized commercially. And then we proceeded to join these together in a YAC vector that was also commercially available. The technique is here. You have your overlapping fragments with your YAC vector, you transform them into yeast, and I should say we bought all these components. About 2 days later, 2 to 3 days, you get your yeast colonies, you screen these via multiplex PCR, so this takes 1 or 2 days, and then you can verify the clones by nanopore sequencing, which we can do in our laboratory. So the whole process, if you have the gene fragments, can take less than a week to get a clone. And then you can transfer your YAC clones into bacteria, which we can do for SARS-CoV-2, but not all coronaviruses, and propagate them if they're stable. So we've managed to do this for SARS-CoV-2. We've also made different reported viruses by substituting different reported genes for different small accessory genes, which are at least can be dispensed with in cell culture. And we've also made these other tools called replicons, where we get rid of some of the genes, such as the spike gene, that allows the virus to propagate, and replace them with selectable antibiotic markers, and include other sort of reporters such as luciferase or neon green. The system we use, we take our back plasmid, we linearize it, we use that to transcribe RNA in vitro and introduce that into permissive cells, and then we're able to recover virus. And you can see an example here just for SARS coronavirus. Here's our virus here, stained with antibody against the N protein. And here's our fluorescent virus that expresses M neon green, and also just detecting the N protein as well. So we've sort of gone on to use this in different funded projects at the moment to look at the variants of concern. And we've produced now a whole lot of different viruses, where we've got the Wuhan backbone with the spike of different VOX, so alpha, beta, gamma, and delta. And also we've got the backbone of these variants of concern, and we've substituted in the spike. So by doing this, it'll allow us to analyze mutations that occur in these VOX and determine whether they contribute to sort of enhanced replication or pathogenesis of these viruses. For our replicon, we use a similar technique. In this case, we don't recover virus. And you can see here, we've got different replicons that have different fluorescent markers. And you can see that our transfected cells show signs that these replicons are stably replicating in the cells. But the problem for SARS-CoV-2, so far worldwide, nobody has been able to then select these cells using antibiotic selectable markers and get populations that harbor these replicons. And that's due to cytopathic effects. However, at the moment, we've got a quite good transfection efficiency, and we're hopeful that we can select some cells that stably maintain these replicons, and then we can use these for antiviral screening. So this brings me to a few points to consider. We can now very rapidly synthesize novel coronaviruses. And one of the sort of blocking points really is the ability to produce synthetic DNA. So we can produce viruses without even having them necessarily in the country. And I think, you know, in the UK at least, we don't have a facility to synthesize large quantities of larger fragments of DNA. And this has really hindered some efforts in the UK. It takes several months now to get these. Obviously, permissive cell lines, just as for the viruses themselves, are required as well. And for feline coronavirus, this has been a limiting factor for us to establish the full virus. These replicon-containing cells are very useful, once again, not having the virus, but we could use them at sort of BSL-2 rather than BSL-3 or 4 conditions to screen antiviral drugs against a large amount of coronaviruses. But for some of them, it's not trivial because of cytopathic effects. And the other question is, obviously, where should the line be drawn when we start looking at gain of function experiments and swapping different genes, either within different viruses, such as we're doing for SARS-CoV-2, we hope in a safe manner, but potentially between different coronaviruses as well, which is another issue. Just to thank my co-workers at University of Bristol, Julian, who's been instrumental in helping to get funding through the FDA for some of this work, Miles Carroll, and also the Genotype to Phenotype Consortium. Okay, thank you very much.